Okay, thank you everyone for taking the time to join me to go through our presentation. Here are the legal disclaimers, which I think you should take some time to look through, so there's some important factors in there for you. So a little bit about us, e-Therapeutics. We have a computational biology platform at our core. We're an AIM-listed company based in Oxford. We have recently announced an addition drug modality called RNAi, and in this presentation I'll go through some of the commercial collaborations we've got with some top-tier pharma companies. So in essence, what do? We construct in silico or computational models of any biological function. So when we look at a particular biology or a function that we're interested in, it can be anything, let's say hyperinflammation in SARS-CoV-2. We then go on to analyze those models to look for novel targets or biological mechanisms. From there, we can then design novel small molecule drugs. And as I said earlier, I'll be going through the RNA platform as a drug modality as well. So you could think of our capability as having an in silico or computational laboratory. So here's a slide giving a lot of different examples of what these models look like, and you can see there's a lot of sort of diversity in terms of SARS-CoV-2, or you can look at things like neuronal autophagy or endocytosis. As I said, almost any biological function. And the models, as you can see, we've generated, over our history, millions of these types of models. They're in essence genes or proteins which if you can think of them as collaborating with each other, talking to each other in a kind of network, and you can see the variety of different networks we have. So this kind of network formation, if you can think of it, let's say within a cell and a biological process of, let's say, as we said, let's say SARS-CoV-2 hyperinflammation, gives that function its resilience and all the things that it needs in order to be able to sort of function. And our modeling is, we think, the truest and fairest sort of representation of biology. Okay, so how are these models generated? Where do these proteins or genes come from? And how do we go about doing this? So as I said earlier, we have a computational platform. You can think about it maybe as sort of 4 or 5 databases or very large data sets or functions. As a starting point, we have the largest and most accurate what's called protein-to-protein, human protein-to-protein interaction database. That kind of is the starting point for us to be able to stitch these proteins together to create the models that we want. The second component of that is we use very modern AI techniques called natural language processing. So we can, if you like, import and extract and save and be able to sort of analyze any kind of paper that's been written, any biological experiment that's been done. So we bring them into our sort of databases and we analyze them. And the kind of third and sort of fourth component are we have also the largest small molecule compound database that we know of. So something in the region of 17 to 18 million small molecule compounds. We have all the approved small molecule drugs, and then a variety of different, in the millions of different compounds. So when you think of the kind of totality of the databases and data sets that we have, there are going to be certain aspects of it which are kind of the data is excellent. So for example, in the approved small molecule compound database, they're obviously very well annotated and known. Some of the compounds are very poorly annotated, and the same applies to the protein-to-protein interaction databases. So kind of feeding into that is where our proprietary machine learning algorithms come in. So there what we're trying to do is fill in the gaps with predictive machine learning algorithms and artificial intelligence, so that where there is data which is not as good as some of the other parts of the data, we can basically create models that again give that sort of true and fair reflection. Okay, so in terms of outputs, again, imagine now we're still in silico or within a computer. The networks, as I said, we can generate, novel mechanisms from that. We can get genetic insights. So we can take genetic data as an input. So think about, these large population studies called genome-wide association studies, looking into very complex diseases like Alzheimer's. Typically the output from those studies are maybe 100, 200 genes, which all have a small influence, if you like, on the totality of the disease. But obviously you can't do anything with 200 genes in terms of actionable outcomes. We can take that information, create our network models of them, and from there we can look at creating drug targets. So in the next slide, and I'll be going through our sort of, if you like, our compounds, which we can derive from our network. So here it's almost like a sort of computer game, if you want to think about it that way. So, we go parallel with the model. So imagine on the left-hand side, we're looking at a model of, again, SARS-CoV-2 hyperinflammation. On the right-hand side, we have our compound database, and we attack the model with every single one of these millions of compounds that we've got. The idea being we want to find the compounds, the highest ranking compounds, which affect the network the most. So typically the output from that is out of the tens of millions that are in the database, we get a very sort of curated list, something maybe like 500 compounds ranked by their sort of activity. Okay, so I guess this slide is really kind of the scoring system in some ways of that computer game or our compounds. On the left-hand side is projects that we've embarked on in-house. So these have been projects which are not just in silico, but we've taken them all the way to in animal models or kind of IND filings. Again, very diverse biological areas. We have 3 partnerships: type 2 diabetes with Novo Nordisk, CNS with MSD, fibrosis with Galapagos. So kind of what you're seeing on the right-hand side, as I said, is the kind of scoring system. So one step back, a traditional kind of small molecule approach would be you set up an assay which says, okay, this is my model for fibrosis. And a typical large pharmaceutical company can really sometimes throw 500,000 small molecule compounds. They literally make them and hit this assay. This is in a real lab setting, and they'll try and see out of those 500,000 how many hits there are. Hits being defined by a compound which has activity against fibrosis. And this is as you can imagine, torturous, long, and expensive kind of process. And the hit rates are desperately poor. Can be as low as 0 or 0.1% of those 500,000 compounds can be active. Then, once you've even got a hit, that's a kind of what's called a blind screening process. So you have no biology knowledge of why this compound is affecting, let's say, fibrosis in these assays. So even with a hit, you have to go back and kind of work out, what's going on, what's the target, what's the mechanism. So from our sort of process, remember, we're coming at it in a kind of biologically aware and relevant way. So we're starting with something we're interested in, then we model it, then we find compounds in silico in a computer, which are a very small number of compounds, so typically 500 to 1,000 compounds, much, much smaller, kind of very curated and precise number of compounds. And our hit rates are, orders of 100 to 1,000 times magnitude to kind of industry standards. So, we do the modeling in silico, we create very, very small number of compounds which are biologically relevant, and that's a materially different process than what goes on traditionally. Okay, so a little bit about our RNAi announcement as to why we have added this, what's our advantage here. I guess this first starting point is RNA platforms are very much sought after, and we'll go through a few slides to show why. And the second component was that we felt that our informatics and our computational platform would be particularly useful in RNA drugs, and I'll go through some of the reasons why that would be. But at its core, we have an in-depth knowledge of the field of RNAi. So really, what would have taken potentially years to develop has taken months because of the kind of knowledge we have. I have to admit, actually, the lockdowns and the ability to be able to really focus on things has really sort of helped that process. And we felt that putting that knowledge and that history of people who've been there and done it before with our computational capability brings us a kind of different seat at the table when it comes to discussions with partners. So I thought it'd be interesting to just, talk about RNA drugs. And on the left-hand side is an example of the current mRNA vaccines, and on the right-hand side is a sort of schematic of our RNA platform. So essentially, all RNA drugs have three sort of bullet points which are the same. Number one is they always have a sort of delivery system. The delivery system carries a genetic sequence. And the kind of important last part is when the delivery system delivers the genetic sequence into a cell, the actual process, if you like, the drug-like effect, is carried out by the cell itself. So in the example of the mRNA vaccine, as many of you will know, the genetic sequence is the genetic sequence for the spike protein. The likes of Pfizer, BioNTech, Moderna will put the genetic sequence, as you can see here, wrapped within what's called a lipid nanoparticle. Upon injection, the nanoparticle basically delivers the cargo or the genetic sequence into a cell. And at this point, the cell's machinery actually reads this genetic sequence, creates the spike protein, and it's the creation of that spike protein which alerts our immune systems to create antibodies and therefore immunity. In our example on the right-hand side, a kind of similar but distinct in one very important way. Our delivery system is on the right-hand side there, this kind of schematic or construct that you can see here. This will ensure that our genetic sequence will go to the liver. And as you can see here, this is our sort of template of our sort of genetic sequence. Our process is slightly different in that we are interested in target genes which are disease-causing in the liver, of which there are something in the region of sort of 10,000. And the first thing we do is we say, all right, this is a particular gene we're interested in. We find out what the genetic sequence for that gene is. We create our own genetic sequence against that gene. Again, delivery system will take it into liver cells and only liver cells. And what happens then is distinct to the vaccines. So in a vaccine, you're creating something. In our platform, what happens is you're shutting down the activity of the gene. So if you like, the kind of left-hand side, the mRNA vaccines or mRNA drugs, they turn genes on. Our platform specifically turns gene off in the liver. Okay, so what were the other commercial rationales that we were, considering? Why did we do this? RNAi in the liver has got some unique drug-like properties. Probably, in my opinion, the most reproducible and validated drug platform that there is out there. And a big component of that is the long duration of action with one subcutaneous injection. So on the right-hand side, you'll see something. Day one here is a very kind of small subcutaneous injection. The target gene in the liver in question is called PCSK9. If we follow the purple line here, what happens is upon injection on day one, you can see the activity of that gene being repressed or shut down. But what's kind of remarkable about it is you can see that this repression or suppression of the activity continues for 180 days, actually goes out to around 240 days, all from one, very simple subcutaneous injection. So, what this platform has really become is it's gone further than just a potential rare disease platform. It has become a potential platform for very common diseases, primarily because of this kind of long activity from one injection. Okay, sorry, I can't move the slide. No, I can. Another important marker here was that, the platform has been validated with a very, very large database of efficacy and safety in man. But more importantly, there are now, over the last 2 or 3 years, there are now 4 approved RNAi drugs on the market. So, in terms of sort of risk and the optimal time to be in this area, this is the right time where a lot of the sort of heavy lifting and failures which can happen in these industries have been done, and a lot of experience and knowledge has been gained. Obviously, financials and financial deals are a very big component of what we do and a very important part of the kind of valuation metrics. You'll see here that in the RNA field, as opposed to, let's say, the small molecule field, in a small molecule program, you're unlikely to do a partnership deal unless you have a sort of probably a phase 1 at least in man, and you show efficacy and safety. In the RNA world, you can do a deal almost as early as day one. One deal I always highlight is Dicerna and Novo Nordisk. So Novo Nordisk went to Dicerna and they reserved 30 genes in the liver, so something in the region of 10,000 there, for a upfront fee of $175 million, and Dicerna also get milestone payments if Novo Nordisk go and prosecute this. So you can do very, very early deals at very different stages, whereas in other drug modalities, as I said, you have to really go a long way down the line to be able to sort of prove that. So a little bit about our RNAi platform. As I said, I think we've got a good IP position. We have filed patents in the last month for the delivery component of the drug. This, what's called position-specific stabilization chemistry, this is the genetic sequence, as you can see, and the colored greens and blues are an absolute essential part of what's called stabilizing your genetic sequence so that the immune system will not be able to attack it, and so the delivery system will be able to transport it to a cell so that the drug-like effect can happen. Without in-depth know-how of how to stabilize these things, it's extremely hard to operate in this area. We've got some sample different permutations and combinations. We're testing to see which one of these we think are the kind of most potent and the ones that we want to use. I guess at this point, it's important to say that the preclinical timelines really are markedly faster than, again, let's say small molecules. So, I can, from here, if I had a target gene in the liver in mind, I can, on my computer, look up the sequence for that gene. I can design my drug, if you like this picture here, within days. I can press send, and that drug will get sent to my CRO. So days to create a drug in silico, if you like. And within a few months and a cost of a few $100,000, I can synthesize that drug, and I'm good to go for studies in animals and obviously in man. So in terms of comparison, by way of comparison, to have a small molecule, even a candidate drug, can take as long as 3 years, maybe even more, to be able to iterate to a good compound. So that's already a huge time difference, and the cost difference is significantly more. So, when some of the sort of kind of valuations and people talking about RNA drugs and being digital, if you like, this is really what they're talking about, is the ability to be able to make your drug not just in a computer, but actually make it, if you like, in chemistry terms, and to test it extremely quickly is very, very different to what you're seeing in other drugs. So as I said earlier, our genetic sequence and the delivery system will take the cargo only to the liver. As I've said, something like over 10,000 genes in the liver, so very, very active organ. Only 1% of those genes is being currently looked at or targeted by any kind of RNA therapeutic, and we think there are many opportunities for us here. Interestingly, the competitive landscape is very, kind of concentrated on a small number of those potential genes, and as you can see by these bars, sometimes 3 companies are trying to target one particular gene. So we think there's a lot of white space in order for us to be able to compete and have a big competitive advantage. So, as I said earlier, the rationale for RNA was in-house expertise and the ability to basically get the toolkit up and running quickly, but also to be able to really differentiate at a kind of partnering level by using our informatics platform. So to that end, over the last really 4, 5, 6 months, we've set up a liver-focused informatics team. We've got the leading experts in liver-specific biology. We really sort of extracted, if you like, hepatocyte or liver cells out of our core platform and any related data, and created a sort of sub-unit, if you like, focused on that cell type and that biology. And I would kind of say, probably today we've got the most complete network map of hepatocyte biology, which is proprietary to us. We're going to go further than that. What are the kind of plans that we have is, things like, let's say there are 10,000 genes in the liver. We want to use our platform to shut down every single one of those genes and to be able to measure the outcome of what's happening in the cell one by one. Almost like creating, if you like, a hepatocyte atlas is what we call it.
1 minute left.
So in terms of collaborations, as I said, Novo Nordisk, MSD, Galapagos, good and very kind of validation of our computational approach and of network biology as a whole and the modeling process. We're actively looking at new collaborations and ongoing business development discussions. So in terms of future plans, we have to expand and invest in our computational platform and data sets and be able to iterate and improve. Big component for us will be this zooming into hepatocytes and really trying to create that, world-leading expertise as the go-to people for that cell type and the potential gene targets for our RNAi platform. And that's our team. And I think I finished. I did it on time, right?
Well done, Ali. Now, unfortunately for me, I sneezed just as you were saying something of great beauty. The phrase that I picked up was the truest and fairest representation of biology. Were you talking about your platform reflecting that or creating it when you were talking about the truest and fairest representation of biology?
Well, so like a traditional way of looking at biology is, I don't know, some of your viewers might know is what's called a pathway. It's a very two-dimensional way of looking at biology. Whereas our way of looking at biology is, we think, in reality, what is happening. Biological functions have to be resilient. Even simple things like eye color, if they weren't, your eye color would be changing all the time, right? So they're extremely resilient, they're extremely complex, and they're in this kind of three-dimensional shape. They're connected to each other to sort of create that sort of complexity. And that, we think, is the real sort of model of biology. And what we're seeing with our partners is they're kind of really embracing that we've got to sort of get away from this for kind of very simplistic two-dimensional or single target way of looking at things.
Understood. I'm glad that we explored that a bit. Now, somebody who's in the room with us, Lillian, is thinking out loud and she's musing. She says, just wondering whether to date, have there been any drugs or medicines developed using these computational techniques?
I mean, they've definitely been part of the process, yes. I mean, in drug discovery, which I think is the area really what she's talking about, we don't also just rely on those. We that's a part of our sort of process of trying to find targets. But yeah, there's no question that people are getting into this area more and more.
Okay, compounds. I'm just looking at an RNS from the 8th of December last year. You had announced the results of experimental testing on the compounds for the treatment of COVID-19. And Andrew Nesbitt was wondering, do partner companies provide the compound libraries?
No, they don't. The compound libraries are basically our own databases. So in the example you gave, it's something called what's called drug repurposing. So, normally what we do in a project is look for novel chemical matter, i.e., compounds which have really not been seen before. In the example with COVID, what we were looking for is using our network biology and our computational models to look in our database for drugs that are either approved or have been through human clinical trials to see, is there anything in that ecosystem with a good body of some safety and efficacy that could very quickly put into man. And we found a drug of a company called Reata called bardoxolone-methyl, scored very highly in our models. We then took them into the lab and it verified what our computational model said, almost by far and away one of the best drugs out there against the SARS-CoV-2 virus.
So just looking at some of the initiatives that you've set yourself since becoming Chief Executive last autumn, initiatives including a scientific advisory board that will help the firm establish your technology in RNA interference. How's that AIM coming along?
Yeah, I mean, that's part of what I was saying about the kind of in-depth know-how within the company. So even, the head of the scientific advisory board was the head of RNA interference at Merck, which was one of the pioneers of what this actual platform is. So, not just with Paul, who's the head of that, but we've had people in the company who've been there and done it in the RNA field, and that's the ability and know-how that I was talking about, to be able to so quickly get up and running. It shouldn't be this quick ordinarily, is what I'm trying to say.
Okay, we're all being far too greedy with what we're wanting from you. But the technology in RNA interference, it could be used to tackle obviously a diverse range of diseases. That's what you're telling us about. Potentially even COVID-19. And to quote you, the inflammation caused by COVID-19 is complex. Network biology can tackle any biological process.
Mm-hmm. Yeah, that's what I was saying at the early part of this presentation. We can go back to the slides where all the networks were. There was such a we're kind of almost disease and biological process agnostic. So from our databases, protein-to-protein interaction databases, and everything else we have, you could come to us with any question, if you like, and we can create models of it.
Okay, so I'm sensing impatience in the chat room. John Unikowski asks, what has happened to your clinical trials program?
Do we have one? I didn't know we had one.
Are you considering it? Obviously, John wants to know.
Well, I mean, look, we're going to try and progress, very kind of high conviction candidates, but I don't think, now we're going back, I'm trying to remember, I don't think e-Therapeutics has really, maybe not for 6 or 7 years, had a clinical trial candidate. I would say at the moment, but not necessarily for that long, this company is in the drug discovery space. Trying to find targets, mechanisms, and to be able to completely differentiate from a very crowded space of looking at the same thing.
Okay, so Tim asks, has there been any new developments in your collaboration with C4X to look at new treatments for Parkinson's disease?
No, not at the moment, no. But we're obviously going other ongoing collaborations where, they're progressing, and we hope to be able to come out with some outcomes in those. Okay,
some final 2 questions. Lillian has noticed that the share price has risen 325% in the last year. How can this be explained? I know a lot of it occurred at the time of your appointment to chief executive. But aside from that, because I've blown your own trumpet for you, why else do you think the share price is rising, Ali?
I think, coming together of a few things, there was some big fund liquidations, which I think we all know about in the UK. Those 2 funds owned 50-60% of this company. So I think the shares were at very exaggerated low prices because of just a big volume of supply. And the second thing, part of it is, I think, when you look at the US biotech market and the biotech index, during the entire sort of lockdown and COVID sort of problem, there's been a material change in valuations, which has no doubt helped us. And yeah, I think, some combination, not just me, but just a fresh set of eyes and blood into the company. I would put those 3 factors probably as the sort of main reasons.
You're a great diplomat. Joel Deacon asks, have you bought any shares?
Lots of them. I've bought, I think I've bought 60 million or 50 million shares, 52 million, something like that. Yes, I've bought lots.
Okay, so you are a former professional chess player. You still play. So in terms of viewing your company as a chessboard, what pieces are you going to play next?
Oh, it's a tricky question. I think, I think we're in 2 very important thematic sections of biotechnology. One is computational biology, which to my mind, and I think a lot of people, there's no question that this merger of, let's say, traditional technology and biotech is coming together. And the second aspect of this is as I described, this kind of digital field for RNA drugs, where you can, it's kind of sequence-based and information-based almost. You're basically, you look things up, you can create drugs very quickly in 3 months. So, the timelines and the data sets are really coming together. So from my point of view, it's to create the best computational models and platform that there is, so that we can basically work with people and ourselves to do things faster, more specific, and the ability to be able to actually do something about that with highly sought after drug modalities, which are extremely fast, at least against traditional drug development timelines.
Okay, final question, I promise. Does e-Therapeutics have the potential to become a major force at this intersection of technology and life sciences?
Well, I'm not saying no, that's for sure, but the answer is absolutely yes. I think sometimes one of our biggest issues is in the UK market, it's kind of almost too novel, there is a very different, as your previous guest asked about NASDAQ listings, and there's a very different kind of philosophy in US investment, but that's retail investors, not even just specialists in timelines. So some of the competitors I've shown in this slide deck are still not profitable 22 years from starting. So I think, capital permitting, this company can be one of the biggest biotechs in the United Kingdom.
And we've run out of time, otherwise I'd be asking about capital. There you go. Ali, you've left us on a cliffhanger. Ali Mortazavi, Executive Chairman and Chief Executive of e-Therapeutics, thank you very much indeed. Don't be a stranger. Thank you.
Thank you for having me. Okay.
Thank you.